Assalamualaikum and good morning. Welcome to another installment of the IMR-SEAMEO Webinar series for the year 2025. I'm Sumarni Ghazali from the Biomedical Epidemiology Unit of the Institute for Medical Research, and I'll be moderating today's webinar. Before we get into the presentations, since our two speakers today are from the Biomedical Epidemiology Unit, allow me to share a quick introduction of this unit, just to give some kind of context to the work that we do and how it relates to today's topic. So, this is the title of our webinar today: Systematic Review of Dengue Molecular Epidemiology, Protocol and Early Findings. So, the Biomedical Epidemiology Unit is one of 4 units under the Special Resource Center at the Institute for Medical Research. At the BEU, we focus on understanding diseases through 3 main areas: disease modeling, GIS, and molecular epidemiology. In the disease modeling core, we use statistical and computational tools to analyze trends and forecast disease patterns and outbreaks, while the GIS, or Geographic Information Systems, or spatial epidemiology core, applies geospatial tools to map disease patterns, identify hotspots to support more targeted public health action. And lastly, the third core is molecular epidemiology, which is an emerging focus for our unit. We aim to bring together genetic sequencing and lab evidence to study disease transmission and pathogen evolution. Using these 3 approaches, our unit aims to support better surveillance, preparedness, and evidence-based policy decisions for the Ministry of Health. Okay, just a quick reminder before we move on. If you would like to receive an e-certificate for attending this webinar, please click on the attendance link in the description box below. Also, our Q&A sessions will be conducted at the end of both our presentations, but if you have any queries, you feel free to send them using the submission link also in the box in the description. All right, let's get started. So today's webinar, as I said, is a systematic review of dengue molecular epidemiology, protocol, and early findings. Before we dive into the actual presentations, for anyone who may be new to the term molecular epidemiology, epidemiology is the study of the distribution and determinants of health and disease in populations, and the application of this knowledge to control health problems, whereas molecular epidemiology builds on this by adding genetic and lab data, integrating traditional epidemiologic methods with molecular techniques. So molecular epidemiology gives insights that traditional surveillance alone cannot provide. All right, now that we've grounded ourselves with what is molecular epidemiology, let's move into the work our team has been doing in this area. We have 2 speakers today. Each will cover a different part of the systematic review. So our first speaker is Dr. Lonny Chen Ahmad. Dr. Lonny is a medical officer from our Biomedical Epidemiology Unit. She has spent more than 15 years working across internal medicine, psychiatry, and research. And now she focuses on computational and molecular epidemiology, disease modeling, and computational psychiatry. Okay, her presentation is on the methodology framework for the systematic review. Dr. Lonny, over to you.
Thank you, Puan Sumarni, our moderator for today. Good morning, everyone. My name is Dr. Lonny Chen. Today, me and Dr. Sarbhan Singh will be presenting our work on systematic review on epidemiology of dengue. We are from Biomedical Epidemiology Unit of IMR. Today, we will walk through through our comprehensive analysis of dengue molecular epidemiology of dengue in Southeast Asia. So I'll be covering the methodological approach, and Dr. Sarbhan will be covering the preliminary early findings. Okay, so now before diving in further into the methodology, I would like to actually lay a foundation and establish what is molecular epidemiology. As Puan Sumarni mentioned earlier, it's an integration of both molecular and genetic tools into epidemiologic research at the population level, meaning it combines traditional epidemiologic methods with laboratory throughput techniques to examine genetic and molecular characterization of pathogen and host. So in essence, it gives us a better understanding of the disease itself, the pattern of transmission and outcome. So for dengue specifically, analyzing this virus genetic sequences is important to actually track serotype distribution, the lineage, understanding the transmission pattern, and also to investigate further the interlinked relationship between the viral genetic characteristic and also the clinical outcome, as well as the severity. So this allows us to answer questions that traditional epidemiology cannot, such as what viral strains that causes the disease, how do they evolve, how do they transmit between populations, and at the molecular level. So this is crucial for targeted intervention. So molecular epidemiology transform our dengue surveillance to more of from reactive to a predictive. So instead of asking what causes the disease or how many people get sick, to how can we prevent the outbreak. So now let's move into the methodology. Okay, so we develop a comprehensive systematic review protocol to analyze this molecular epidemiology of dengue in Southeast Asia. So what is a systematic review? Systematic review is actually a robust and also it's transparent and reproducible kind of methods of analyzing, evaluating, and finding certain topics of field. So it's all available research evidence on the specific topics. So with minimal bias. So this can be also time consuming and also resource intensive. So this wasn't just a simple literature review. So it is a robust kind of research review and also transparent and reproducible that follows international guidelines. So you might be wondering why we invest so much of effort in studying molecular epidemiology of dengue. So first is to track viral evolution and circulation, meaning we need to monitor all 4 serotypes that is circulating to actually for effective vaccine development and to detect new emergence of dangerous strain that could possibly contribute to an outbreak. Second, to understand disease severity. So molecular epidemiology helps us to identify what viral genetic factors that causes severe or mild dengue. Okay, and to determine why certain individuals are more susceptible to severe dengue. Third is to understand the disease transmission. So from here, we can estimate the transmission probability, the basic reproduction number, R0, to model an outbreak, and also to trajectory of the outbreak more accurately. Fourth is to actually map the geographic spread, meaning to actually track transmission between regions or countries, allowing us to predict an outbreak pattern and also the epidemic that might occur. So all of this actually leads to an improvement of prevention and control. Okay, so as one said, molecular epidemiology actually provides insight into the causation, pathogenesis, and transmission of infectious disease, leading to a more effective and effective prevention strategies. So why do we actually choose Southeast Asia? is because Southeast Asia bears about 70% of the global dengue burden. This is actually stated by WHO. It is hyperendemic in this region, and it serves as a hotspot for viral evolution and dissemination. So understanding molecular epidemiology is actually important here, and it has a global implication. Okay, so before we proceed further, I would like to actually clarify on the conceptual framework that we are using. So molecular epidemiology research actually organized into distinct domains. So specific areas of investigation addresses specific areas of infectious disease. So these domains are the structures for our methodology and also our finding that we will actually explain further shortly. Okay, so every good systematic review starts with a clear question, research question. So ours was, how can molecular epidemiology be applied to inform and strengthen public health measures for effective dengue control in Southeast Asia? So this question was made intentionally broad because we would like to actually capture the full landscape of molecular epidemiology research in Southeast Asia, from causation to prevention across all the 11 countries of Southeast Asia. To be specific, we actually wanted to examine the evidence across the 7 critical domains of dengue molecular epidemiology and to formulate evidence-based decision that can strengthen the public health strategies. So, to answer this question systematically, we followed the PRISMA guideline. So what is PRISMA guideline? PRISMA guideline is actually a preferred reporting item of systematic review and meta-analysis. This is a gold standard that is used for conducting a systematic review. So our search was actually comprehensive and was based on 5 databases: PubMed, Scopus, Wiley, Cochrane Library, and Embase. So our time period was actually from inception of time to 2024. So this allowed us to actually see decades of research that allows us to actually see the temporal trends and historical shift. We limited this search to English language for practical reasons, and our geographic focus was on the 11 Southeast Asia countries, so including Malaysia, Brunei, Indonesia, Singapore, Thailand, Myanmar, Laos, Cambodia, Vietnam, and Philippines. Okay, next is our inclusion criteria. We ensure that all published studies reports on these 3 domains: dengue, the 7 domain framework, molecular methods, and we exclude article published in grey literature. So next is our search terms and concept. So our search strategy was based on 3 key concepts and Boolean operators. So our first key concept was actually molecular epidemiology. These terms include such as genotype, serotype, phylogenetics, phylodynamics, Bayesian coalescence analysis, and so forth. We cast a wide net to capture all molecular methods so that we don't miss any methods. And the second concept is dengue itself. So we use obvious terms such as dengue, dengue fever, as well as clinical presentations such as dengue hemorrhagic fever and dengue shock syndrome. The third key concept was on the geographic focus of the Southeast Asia country. We actually use regional term as well as the 11 individual countries' name. So by combining all of these 3 concepts and the N operators, we actually extensively captured all the studies that addresses the 3 inclusion criteria, 3 domains in inclusion criteria. So next, so this is the most critical step in any systematic review. That is the quality assessment. So we didn't just choose randomly or choose or include any studies that we found. We actually vigorously evaluated one and each of them. So to ensure the robustness of the evidence synthesis, we actually use two validated tools. First is the NOS, Newcastle Ottawa Scales, for observational studies, and second was JBI checklist. This is for different study designed. So first, the NOS checklist. It's based on the scoring system with a scoring maximum of 9 on 8 items questioned. So with more than 7 is actually high quality and less than 4 is poor quality. So it has several domains. The main 3 domains are on the selection domain, comparability domain, and outcome or exposure domain. For the JBI checklist, is more focused for different types of studies, and each of this checklist has at least 8 to 13 questions. It's more of yes, no, unclear, or non-applicable kind of questions. It assesses on the selection bias, measurement validity, statistical method, and also confounding controls. So it is based more on the percentage based, and if more than 70% is actually high quality. So all this in general actually assessed the study bias, the study design, risk of bias, reporting completeness, and molecular lab or standardized lab protocol quality and validity. So the quality distribution is if more than 70% for JBI is high quality, and if less than 50% is poor quality. But for NOS, more than 7 is high quality and less than 4 is actually poor quality. So all this brings to, all this actually is to ensure that our findings are on high quality and not on based on volume of publication. Okay, after the quality assessment, we will go through the next stage or next phase on data extraction. So data extraction is actually systematically pulling all the key details from each included study to their respective domains. So this standardized method or standardized extraction actually to ensures that all the details or the data that we can synthesize finding across all the studies comes out in a meaningful way. The next step, we proceed with data analysis. So during the data analysis, we actually have descriptive analysis and analytical analysis, and we use mainly Stata and R software. So, okay, so this Prisma comes into a flow chart. Okay, so I will walk you through the steps in another next few slides. So the first phase was actually the selection process. So we start off with an identification phase. So as you can see that the studies are all extracted from these 5 databases, and we started with first hit of 8,197 articles across all 5 databases, in which Scopus has the highest number of 3,758 articles, and the smallest number was actually from Wiley, 44. We actually extract all this data and use EndNote as a citation manager software. Subsequently, we screen it with Ryan and remove all the duplicates. So we had about 4,390 records after the duplicates removal. We then actually applied inclusion-exclusion criteria to it, and we actually comes to removal of 4,023 studies. So there are various reasons to it actually, and one of them, and a few of them, the main important one was actually those article was not doesn't use a molecular EPID method, and the article was not in domain, even the setting was not right, it was not in Southeast Asia, wrong publication, and we did not also receive full text, and so forth. So all this comes to a number after the screening was 367 included with full text, with the highest number of cross-sectional study. Okay. So this left us with 367, and we found that from that, after the quality assessment, 80% of our study was high quality. So this gave us to this high proportion actually gave us confidence with our findings. Okay, so now we have arrived to our organizing framework, the 7 domain framework. This framework actually shows us the structures on how we actually proceed with our analysis and also our findings. Okay, first we start with the causation domain. So this domain actually explains more of what serotype or what genotype that causes the disease. The second domain explains more of the strain dynamic. So how different strains move and changing along the way, along the time. So it tells more of historical shift of the viral and the disease. Third is pathogenesis. So this is the disease mechanism. So it tells you how the dengue causes illness at the cellular level. Fourth, sources and reservoir. It explains more of where does this virus come from, what are the vectors involved, is there any reservoir, is there any other reservoir to this disease. And fourth domain, the fifth and sixth domain are always interrelated, the drug treatment and vaccine development. So molecular EPID will actually establish more of knowledge to the antiviral development, and also it gives a genetic information to guide the immunization development as well as strategies. Last but not least is the transmission domain. That is the largest domain. It tells us how does this virus spread at the molecular level, and is there any molecular event that's involved to facilitate the transmission, such as mutation, recombinant event, and so forth. So this is our publication for this year. We actually submitted a publication to BMJ. You can have a look and scan it there. Okay, so this completes part 1 of our presentation. So we actually covered what molecular epidemiology is and why it matters to dengue. We also explained our comprehensive systematic review following PRISMA guideline, and also our 7 domain framework. So in infectious disease, knowledge is prevention. So molecular epidemiology works as knowledge to prevent an outbreak that is going to occur or going to start. So this is important to transform our approach from a reactive to predictive to broad to precision and descriptive to mechanistic. So in our region, bearing 70% of global burden of dengue burden, our systematic review will actually provide evidence base showing that molecular epidemiology is essential for prevention and control in Southeast Asia. So at this point, I'll hand over to Dr. Sarbhan, who will actually present the early findings across these 7 domains. Then here you will be able to see what does decades of molecular epidemiology research reveals about dengue in Southeast Asia. So as a methodology, framework is our recipe, and the findings is actually our meal. Okay, over to Puan Sumarni. Thank you for joining us for our webinar today.
Thank you, Dr. Lonny. All right, if you have any questions for Dr. Lonny regarding the presentation, please feel free to submit them using the link as mentioned, and hopefully we'll be able to address them in the Q&A session later. Okay, next we'll move on to the second part of today's webinar. Our next speaker is Dr. Sarbhan Singh, public health physician, and also he's the head of the biomedical epidemiology unit. His research interest is in disease modeling and epidemiological studies. Okay, so Dr. Sarbhan will be presenting the preliminary findings on the molecular epidemiological landscape of dengue across 7 critical domains. Over to you, Dr. Sarbhan.
Hi, good day everyone, and thank you so much Puan Sumarni for that introduction. So I would like to continue our webinar today, where I think at this point all of us, or all of you here would have seen the amount of extensive methodological processes that was applied as described by my colleague Dr. Lonny in formulating the design of the systematic review. And now I would like to, in fact, I'm actually very excited to be able to share with all of you here today some preliminary findings that we have obtained with regards to these 7 domains from our systematic review. So let me dive straight into this. So I'll be going through each one domain or few of them individually, and we can together see what are the findings that we have obtained based on the 365 studies that have finally been included in the current review. So moving to the first domain, this domain is the domain that looks onto actually 2 domains. We clumped it into one, which is the causation and circulation. So in here, we can clearly see that the ones in blue are the variables that we already have some preliminary results, which are basically the dengue cases that were positive by molecular methods, and serotype and genotype. Before I dive deeper into this, for those who are new to molecular epidemiology, what is serotype and genotype? I'll just take this few minutes to explain. Serotype, when we speak about dengue, is basically how the antigen expression varies across the viruses. So in essence, there are basically 4 serotypes for dengue: serotype 1, 2, 3, and 4. Whereas, what is genotype? Genotype is basically how the genetic characterization or expression of these genes vary for each particular serotype, right? So let's dive into this straight. So what is this graph showing? Very simple. This graph is basically a graph that provides information from the year 1964 until the year 2023, which we have data on. So the red plots that you see here are basically the reported dengue cases. And these cases may be either reported using molecular or non-molecular methods. And the line that is dotted here are basically the number of these cases that actually has been confirmed using RT-PCR methods. So what we are trying to see here clearly is that in more recent decades, we can clearly see that this, the number of cases that are actually being confirmed using molecular methods is on the rise. And this doesn't surprise us, because with more recent advancements available in resources, we are able to test more. Moving on, what we see here is that we do see that there is traditionally a cyclical pattern when it comes to surges in dengue cases, and we can see that occurring roughly maybe every 3 to 5 years, as indicated by these blue arrows that you see in the graph here. Now, this is where things get much more interesting. So what I would like to show you here in this diagram is you can see that how different serotypes of dengue, so we have got the yellow one in this plot indicates serotype 1, and the green is serotype 2, and so on and so forth. So from the period of 1964 until 2000, so from the data that we have obtained, what we can see here is that you see that the green area is much more larger compared to the other areas. So this tells us that during this period, it is the dengue serotype 2 that is predominantly circulating. Now what happens as we move forward from the year 2000 until 2008? What we can see here, we can now see that the yellow area is getting bigger. And what does this mean? This is basically telling us simply that we are now seeing a change or a shift from serotype 2 to serotype 1. And as we move further in time, we can see that serotype 1, which is the yellow area, remains the most. So there is no serotype shift here, but it remains persistent. And as we move further in time from 2014 right until 2022, now what we are seeing a decline in the serotype 1 that was predominant, and we can see that this red color area is increasing, and this corresponds to serotype 3. So from our review across the 11 Southeast Asia countries, we can clearly see with respect to time from 1964 right until 2002 how the predominant serotype were at certain intervals in time and how they shift to another serotype, or maybe how they persistently stays the same. Now, that was about serotype. Now here, we move on into how each serotype, remember I've mentioned earlier the dengue serotype, we have 4. Now from our review, we were very much interested to see within each serotype, what is their genotypic characterization. And what did I mention about genotypic earlier? It is basically, in simple terms, how the genetic characteristics differs within each serotype. So if we look at this chart here, let's just look at each serotype. Let's start off with serotype 1 first. So what we are seeing here, across time, you can see many, many colors. Each colors indicate a specific genotype for serotype 1, right, as we can see here. And what are we seeing here? In simple terms, we can clearly say that looking at all these colors at a snapshot, we can say that for serotype 1, it is the genotype 1, which is the red color one, which is kind of persistent or most predominant. All right, that is the first thing we can conclude from here. The second thing we can say is that initially we get a sort of a mixed picture or a co-circulating picture. That means we have got an alternating pattern that is occurring in serotype 1, genotype 1, then switches to genotype 4, and then there is a co-circulating pattern, and subsequently we tend to see a stabilizing pattern. Now, the reason why I am highlighting this is because there is a clear explanation of how these variations of genotype within a serotype would have with regards to the disease transmission, which I will emphasize in a little later. So now when we look at dengue 2 serotype, so we can see in this case it is very, very different from dengue 1. How? In dengue 2, we see that we can see that there are many, many colors, right? And the predominantly occurring colors, or which is the genotype, is basically we have got the cosmopolitan and the ASIA1. All right. So what we conclude here is that throughout this period, there tends to be a co-circulating genotypic pattern when it comes to serotype 2, unlike serotype 1, which had only the red one, which is genotype 1. Now, moving forward, sorry, I got to go back to, okay, moving forward to dengue 3 or serotype 3. Here we can see initially there is a co-circulating pattern which is very, very diverse, and subsequently it tends to localize or focus on just 2 colors, which is the red one and the light pink, which is genotype 1 and genotype 2. And finally, when we look at serotype 4, this is much more stable in terms of its genotypic expression compared to serotype 2 and 3, where it is most oftenly sticking with the genotype 1, right? So now, at this point in time, what information we have? Number one, we had described how the serotypes patterns were in time, and within each serotype patterns, how does the genotypic characterization occur? Now we take those 2 pieces of information, we want to put it together to try to explain periods in which we observed high dengue cases. So let me start first with the period from 2000 to 2010, as indicated by this blue highlighted area. So during this period, we do see spikes in dengue cases occurring. And we now try to explain why this occurred at the molecular level. So let us see, during this period, what this picture is showing. Let me start with this picture first. During this period, we start with serotype first. What we see happened here, we see that during this period, there was a clear shift between serotype 2 to serotype 1, right? And in addition to that, we can see within serotype 1, there were many genotypes that were occurring. Genotype 1, genotype 4, and a co-circulating pattern. So what does this do when we have a change in serotype and we have within that single serotype, which is serotype 1, there is a very diverse or mixed pattern of genotype. So what does this basically mean in layman terms? In simple terms, because of this variation and this diversification, this creates a pool of population who is more susceptible and who is more immune naive to the current recent circulating strain of dengue virus, the serotype, all right? And therefore, more people will be having a higher susceptibility to get infection. Now, if we focus on the next spike, which occurs in 2012 and 2014, so here, interestingly, we don't see a serotype change. That means serotype 1 remains predominant. Now then, why are we actually seeing a rise in cases? Well, let's look at the genotypic expression for this period for serotype 1. And we can see that in the early phases, we can see that despite there being a fixed or a constant serotype, which is serotype 1, it had 3 co-circulating genotypes. So once again, when you have variation in your genotype within the same serotype, it still creates a pool of which people are not are still susceptible towards the infection, and this explains this area, right? And finally, when we move to the other areas which shows a spike, we can see that similar to the first poll, here is also showing that there is a shift from serotype 1 to serotype 3, and we have a co-circulating genotype within each, within the particular serotype, similar to the first plot that we saw. Okay, so basically what I'm trying to get at here is that our findings at this point in time makes it very clear to explain that particular spikes in cases that we are seeing from this study can be explained by serotype shifts, and within each serotype, the variation in the genotypic characterization. Now, let me dive to pathogenesis. So here I'll just be looking at virus genetics. So this is another interesting finding. So here, what we found is that we identified that out of all the specific, so we can see this has been classified by all the 4 serotypes, and within each serotype we are trying to describe where is the area within the virus that we see the mutation most frequently being reported by these studies. And what do we see? We see that the envelope region of the virus is, which is indicated by this dark blue area. This is what most studies report across all 4 serotypes. And this doesn't come as a surprise because it is the envelope area that is frequently coming in contact with the human body, and this is due to immune pressure resulting the envelope areas to have a higher degree of mutation to enable infection to actually be established. Now, then we looked to see, aside from now where the mutation is occurring within the virus, which site, we want to see what type of mutation is most commonly reported. And we zoomed at about 4 to 5 types of mutation. And what we are seeing that substitution is the most commonly reported mutation across all 4 serotypes. All right, of course, these are the most glaring ones that we are seeing, okay? And why substitution, why do we observe this? Well, there is evidence that supports the facts that substitution tends to be the most simplest form of mutation, and therefore it occurs very frequently. Okay, and this helps in the viral replication. And now, putting them both together, the site of mutation, the type of mutation, now we want to see whether these mutations actually in turn do have an effect on the amino acid change, because this is where it's important. There can be mutations, but some mutations may not result in any form of change in amino acid. So here, from our review, we have identified that most of the mutations that have been reported actually do have an effect on a change of the protein, the amino acids, which is the building blocks of the protein. Now, this is concerning because this could in turn affect the infectivity or the transmission of the dengue virus. Now, this is just to summarize basically how those 3 elements can potentially result in a higher degree of immune susceptibility and higher dengue transmission. Now, I'm moving forward to the 4th domain, and here I would just like to descriptively show some of the sources and reservoir, and I think this does not come as a surprise. it's very obvious that if we speak of reservoir for dengue, mosquito is the most common one found, and followed by humans. But what's interesting here is that there are also some non-human reservoirs that has been identified. And this is important because if there are other forms of non-human reservoir, it's important to identify what they are, because they could potentially also assist in transmission of disease to the vector and subsequently to human. And when we break down by serotype, we are seeing a similar picture where within each serotype, we do see mosquito being the most common type of reservoir, but the non-human reservoir here that has been identified from our papers were reported specifically for serotype 2 and serotype 3. Now, moving on to the vectors, once again, nothing surprising here. It is Aedes aegypti, Aedes albopictus, and we have got several other forms. And this is consistently seen across all 4 serotypes. Now, coming to the 5th and 6th domain, so here, basically, at the preliminary level, what we would like to share is to see what are the types of drug classification that have been reported either to treat dengue across different countries. So here we can clearly see that we have a combination of drugs that are being formulated naturally, using naturally derived products, and of course non-natural. And this group of drugs, basically most of them work on an antiviral property, with the second line being more of an immunomodulatory effect. Now, and then moving on to vaccines, well, at a quick snapshot, we can clearly see that from our review, the most commonly reported vaccine against dengue is under the group of Dengvaxia and Takeda, while many other studies have reported on precursors that can potentially be used next time for vaccine development. Now, finally, I move to the last domain, which is the domain on disease transmission. All right, so mode of transmission here, basically we can see that yes, basically dengue is a vector-borne disease. So we know that it's transmitted by vector, which is the dengue mosquito, right? So, which is, you can see, very obvious. But here we do find certain studies that have also reported that there are evidence of vertical transmission, mother to baby. All right, and this has been reported for serotype 1 and serotype 2 specifically. Although the numbers are very, very small, there have been studies that have reported this. So maybe this is an area which we need to explore further to see if such transmission are actually increasing or substantially being reported, because then we can eventually see if there are other routes of transmission compared to the conventional vector-borne transmission method. Now, basically those were the preliminary findings that we have across all 7 domains at the current moment from the 367 studies that we have retrieved in this review. So at the current moment, at the preliminary level, how does this help in terms of public health? Well, for one, we look at it in terms of how we can actually use these molecular data to support or to assist dengue early outbreak predictions or early warning systems, as we know that we would love to have a dengue proactive type of system in place. So what we see here is that, as I've told you earlier, serotypes, genotypes, probably mutations. So these are all, we can consider them as variables that we can include in our outbreak prediction systems, okay, in existing to additional data, which we have case data and things like that, to provide predictive capacity, because we do see clearly, even in this review at a preliminary level, that when we see some serotypic shift or genotypic diversification, this tends to occur much earlier before we see cases start to rise up. So there's a lag. So monitoring them closely would definitely add value in our early warning systems. And finally, we also believe that such information, such molecular-based information, could be very useful when we want to formulate vector control activities, targeted vector control activities. Maybe different serotypes and genotypes would probably be more sensitive to certain types of vector control activities, which could be a potential area to delve further deeper into. As we know that the disease transmission for dengue is not uniform. It's very, very difficult because of the variation in serotypes, and within each serotype, if we tend to have a very diverse genotypic expression, this makes it even more challenging. And put that together with mutations, it adds to a very complex, dynamic, evolving picture of dengue. That's why the transmission is not uniform. All right, and therefore at this current level, these are some preliminary findings that we have based on the review, and we are still, actually the review is still ongoing, so there are a few more areas to get findings on. At this current stage, we are quite happy to share what we already have. And with that, I would end my part of the presentation, and I'll pass it back to you, Puan Sumarni. Thank you.
Thank you, Dr. Sarbhan, for the interesting and thought-provoking presentation. Okay, so both our presenters have presented their respective methods and findings. So now we open the floor for questions and answers. If you haven't submitted your question, you can still submit using the link we provided in the description box. We will try to cover as many questions as we can. Okay, so I have one question for Dr. Lonny, which is why did you focus on Southeast Asia?
Thank you for the question, Puan Sumarni. This is actually an excellent question. So as from my previous slide, I did mention that dengue is hyper-endemic in this region with the highest global burden. 70% of the global dengue burden, okay, this is stated by WHO, but it's also because of the unique regional characteristic. It has all four actively co-circulating serotypes, and then we also has significant cross-border movement from traveling, trades, and even share same vectors. And our regions actually has diverse climate, urbanization, and even public health system. So, but yet sufficiently similar for a meaningful regional synthesis. So this actually provides a regional insight, relevant insight to actually that may not be able to see if from global data. So findings are more actionable for Southeast Asia public health authorities actually. So that's that. Probably we also considering expansion to global comparative analysis for this study as well. Thank you.
Okay. All right. And now, one question for Dr. Sarbhan. Does the 3 to 5 year cyclical pattern you identified correlate with serotype shifts?
Right. Okay. So this question is asking whether in every time where we see basically the presence of either serotypic change or genotypic change, does it in turn cause a spike in cases? So if we look at what we have been traditionally observing, especially the first few slides that I have shared with you earlier, there are evidence to support that when there is a serotype change or a serotype shift, we do anticipate or observe a rise in case, because as I've mentioned earlier, there would be more immune naive people. What do I mean by this immune naive? It's basically very simple. If you got a group of population where dengue serotype 1 is frequently being circulating there, that means this group of people would tend to be, their immune system would tend to be familiar with the dengue 1. But suddenly now you throw in dengue 3, the immune system will take time to react to that. So infection to dengue 3 will flare up. However, having said that, this is not always the case. If we don't necessarily see a serotype shift, but we do see a spike in case, this can also possibly occur because, as I said, within a particular serotype, there could be many, many different genotypes. So if there is a change in the genotypic characterization, then that also could in turn result in increased transmission despite having the same serotype. So we are observing this pattern. Of course, we cannot say this as a rule of 100%, but these are building blocks of evidence that could support a change in serotype, or even within the same serotype by having genotypic diversification, it could result in an increased number of transmission. And that's why I believe it's very important that we include these variables as parameters in dengue early warning systems. So yeah.
So in short, this can be used for outbreak prediction, is it?
Yeah, I mean, that is actually a goal that we should work towards, because in integrating and incorporating molecular-based evidence into dengue predictive models or early warning system is actually an area to go to right now, because they do provide information that has the potential to predict. I wouldn't say predict, that would lead to an increased disease transmission with the lag, especially as what we are seeing. So yeah, I mean, that's something that is very much an area that we should start to explore.
Okay, and one last question for Dr. Sarbhan. You showed in one of the slides that the virus envelope protein has the highest frequency of mutations. So what are the public health implications of this, if any?
I mean, from a public health standpoint of view, whether okay, so at this level, right, if we see the envelope area being the area where most amount of mutation tends to occur. So at a very superficial level, well, one thing that we can draw from here is when efforts are being made to formulate or develop drugs or maybe even vaccines, this could be an area where these drugs or vaccines should predominantly target, since it's the area that is always evolving and changing. So this is one area from a public health standpoint of view, and also maybe from terms of surveillance. Routinely monitoring mutations across the viral structure could also give us an indication where we see that if maybe it's still sticking at envelope, what should we do? Or if it starts changing to maybe another structure within the virus, it could alert or signal something else. So yeah, in terms of public health, I would at this current moment say probably for drug and vaccine developments, as well as in the sense of surveillance, disease surveillance.
Okay, so molecular epidemiology has far-reaching implications into drug development also. Okay, so back to Dr. Lonny. Regarding the framework, how did you decide on this 7-domain framework?
Okay, thank you, Puan Su, for the question. That is an interesting methodology question. Okay, our approach was a hybrid approach. We actually adapted it from a molecular epidemiologist's foundational framework, and also it combines from WHO dengue surveillance that works around etiology, transmission, and control. This framework actually based on grounded question of the molecular epidemiological domains that I've mentioned earlier, the 7 domains: causation, pathogenesis, circulation, transmission sources from reservoir, prevention and drug treatment. So we applied this framework to our 367 articles from bottom-up extraction and classified this study according to the domain. So most of our studies have multiple domains. So this framework actually ensures comprehensive coverage. And we know that every dengue molecular epid studies at least fits one of the domain. So thank you for the question.
All right. Okay, one last question for Dr. Lonny. Why were those databases, the 5 databases, selected?
Okay, thank you, Pansu. Okay, we searched the major databases strategically to cover comprehensively for our searches. So PubMed was actually a primary biomedical database, essentially covers health research and it has indexes journal. Scopus has the largest abstract and also citation database. It was also chosen because it covers more of Asian journals. So this is vital for our Southeast Asia coverage. MB is particularly strong in Europe and it focuses on pharmaceutical and drug journals. So this actually is important for our drug and treatment. Cochrane Library is actually a gold standard for systematic review and trials. So essentially it covers our vaccine domain. And last but not least, the Wiley, actually it just adds up coverage journals that are maybe not fully indexes. So all this actually a multidisciplinary coverage with geographic balance lah. So at the same time, we also use snowballing methods to actually covers to catch anything that we miss that is actually not in our databases searches. So as you can see from our presentation earlier, Scopus was as the highest number. It's about 3,700 plus. So we confirmed that most of our article was extracted from Scopus was actually of Southeast Asia journals actually. Thank you so much.
All right, it seems like that's all the time we have today for this webinar. So as this is our final IMR-SEAMEO webinar for this year, we would like to extend our thanks to all our long-time followers of this series and also to those who just joined for the first time. Thank you all. We appreciate your support and we look forward to seeing you again next year with even better science to share. Okay, take care, stay safe, and Assalamualaikum.
Thank you.